Hello, everybody. Today we have lecture second, and it will be about morphological and ultrastructural peculiarities of Actinomycetes, Spirochaetes, Rickettsia, Chlamydia, Mycoplasma, and fungi. Okay. In the lecture, in the first lecture, we talk about bacteria at all. But you should know that there are some bacteria that have difference and they are not look like another bacteria. That's why we need to another bacteria. That's why we need to talk to discuss this bacteria separately. So Actinomycetes, Spirochaetes, Rickettsia, Chlamydia, and Mycoplasma. All of this group have its own peculiarities. They are different from another bacteria. Let's start with Actinomyces. When Actinomyces were observed in first time, scientists they thought that they're fungi because they have morphology very similar to fungi. But when they studied it more, when they studied the morphology of actinomycetes structure, ultrastructure of their cell, it became understandable that actinomycetes are not fungi because they have prokaryotic structure of the cell. That's why they belong to the order Actinomycetes and family. There are two families, Actinomycetes and Streptomycetes. Genera Actinomyces and Streptomyces. And it's important that Actinomycetes that belong to the Streptomycetes, they are used in the antibiotic production because they are natural producent of antibiotics. They synthesize the substance which can act as antibiotics. And what about actinomyces? Actinomyces, they are causative agent of dermatomycosis. They infect tissue from interlaced hyphus druses which are calcified in central. What about morphology? Actinomyces, they slight branched root having flask-like thickening localized at the end of the cell. And streptomyces strongly branched trees, hyphae. They look like a tree. This is actinomyces and this is streptomyces. Actinomyces, they have a hyphae. They look like a tree and they create something uh, uh, look like mycelium. Okay. Streptomyces, they can form exospores and they use this exospores for multiplication because in the lecture first we talked that bacteria they use spores for surviving, but not streptomyces. They are different. They use their exospores um, for multiplication, for reproduction. Ultrastructural feature is usual for prokaryotic cells, so they have peptidoglycan of the cell wall, and also they contain unusual sugar which are not present in other prokaryotes. Okay. Okay, that was about Actinomycetes. The um, and the next will be about Spirochaetes. Okay. Spirochaetes, they belong to the order Spirochaetales, family Spirochaetaceae, and there are three genera: Treponema, which cause syphilis; Leptospira, which cause leptospirosis; and Borrelia, which cause borreliosis. One of the peculiarities is about its shape. Their shape is curling. And second peculiarity is that the flagellum is inside the cell, but not outside as usual for bacteria. Look at this picture. So here we can see Treponema, Leptospira, and Borrelia, and they are different between each other because they have different shape, but all of them are curling. Ultrastructural peculiarities is the organ of motility is an axial filament which is localized in periplasmic space of the cell wall and situated along the cell. The filament is built of contractive protein flagellin, the same as flagella. Therefore, spirochetes move by the way of contraction of the body of bacterial cell. So look at this picture. Here we can see spirochetes, and we can see this is filament. Filament, it's a structure with this structure which is made by uh, specific protein flagellin, and flagellin, it's a protein is uh, which made flagella. That's why we can make a conclusion that that this filament is also flagella, and it is necessary for moving, and but it is not outside; it is inside the cell, and there are specific structure which is called blepharoplast, or this is place of untouring of axial fibrils. When fibrils are stick, they are stick here, they are close to this place. This is the end of this filament of this axial fibrils, and also it has something like cell wall and protoplasmic cylinder. It's something like cytoplasmic cell, cytoplasmic membrane. Okay. And third peculiarity is that spirochetes, they very worse to be stained. So they not sensitive for staining. That's why we usually can't use light microscopy. That's why the best way of microscopy will be dark field microscopy without staining, with where we can see bacteria, alive bacteria. Yeah. They're moving and like that. Borrelia, it's one of the spirochetes which can be stained, but also its staining is very worse. It's not so good as as it is as a bacteria. Mm. Okay, and next group is Chlamydia. Principal difference between them and other prokaryotes is that chlamydia are obligate intracellular parasites. They need live cells, they need a eukaryotic cell to multiply. That's why they are very similar in this feature to the viruses. Viruses also, as you know, can't multiply, can't reproduce without without eukaryotic cell. And Chlamydia also, they can't reproduce by themselves. They need the source of energy, and that's why they use live eukaryotic cell. They belong to the order belong to the order Chlamydiales, family Chlamydiaceae, genus Chlamydia. And also about Rickettsia, they also obligate intercellular parasites. They belong to the order Rickettsiales, family Rickettsia, and genera Rickettsia, Coxiella, and Rochlimae. The morphology, the shape of bacteria is cocci, and Rickettsia is coccobacteria or coccobacilli. Coccobacilli, and Chlamydia are the shape is cocci. And they need live cell to reproduce themselves because Chlamydia, they can't produce energy by themselves. They are energetic parasites. And also Rickettsia, they also can't produce energy. They can't produce some energy substance for getting energy. That's why they use energy source of the eukaryotic cell. They multiply in the cytoplasm of the host cell. And they situate like a cytoplasm inclusion, and that's why we can see them in the cytoplasm of the host cell. Ricketts says they diffusely in the cytoplasm or in nucleus, but Chlamydia, they make something like endosome, like inclusion borders, microcolonies covered by the cord having origin from the host cell membrane. To observe this bacteria, it is necessary to use Romanowski-Jimse staining or Stradowski staining, and then use light microscopy. Next, bacteria, which is different from another bacteria, are mycoplasmas. Mycoplasmas, they don't have mycoplasma. They don't have cell wall by its nature. So it's natural to not have a cell wall for mycoplasma. That's why they don't have the shape. They are pleomorphic, polymorphic bacteria, and their cell membrane, cytoplasmic membrane, is contain of sterols to be more suitable for defense, for example, more for the force. That's why its cell membrane is not similar to the cell membrane of the other bacteria, because they lack of the cell wall and they need to use their cytoplasmic membrane as a main shell. And also they have different genome composition. They belong to the phylum Tenericutus, class Mollicutes, or the Mycoplasma thales family, Mycoplasma tatsel, and there are two genera, Mycoplasma and Ureaplasma. Usually they are associated with urogenital infection and some infection of the uh, respiratory tract. Mycoplasma pneumonia, they are associated with pneumonia, of course. We can't use light microscopy because without without cell wall bacteria can't be stay in it. That's why we need to use some methods of microscopy which observe bacteria without stain, and then phase contrast microscopy and electron microscopy will be the best. Okay, so we finished to discuss bacteria, and we start to discuss fungi. Classification and taxonomy. Fungi belong to the eukaryotes kingdom. There are three domains, and one of them is eukaryotes domain, and we have such division as a Kendomycota, Myxomycota, and Eumycota divisions. And pathogenic fungi belong to the next classes: Zygomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Zygomycetes, Ascomycetes, and Basidiomycetes, they are fungi which have sexual reproduction, but Deuteromycetes, they are fungi imperfecti, which don't have sexual reproduction. So first three classes are fungi perfecti because they can use sexual reproduction, and Deuteromycetes, fungi imperfecti. Please remember this moment, notice this moment. Here you can see the cell of the fungi, and they are eukarya. That's why they have uh, all organelles or inner structure which is necessary for eukaryotic cell. Of course, they have nucleus with nuclear envelope and nuclear pore, and they have specific ultrastructural organelles like complex Golgi, like ribosomes, like endoplasmic reticulum, and also they have a centrosome for division, and also they have mitochondria to get an energy, and also they have lysosome to destroy nutrients, and all of them. The fungal cell wall have a difference. It contains polysaccharides present by chitin as structural components of fungal cell wall, which differs from chitin of arthropods, contain less amounts of some substance. And also they contain glucans and mannans, and their cell wall contains chitin. That's why they have some similarities with animals, but because of chitin. Structurally, fungi exist in two morphological variants. Fungi which produce long branching filaments are multicellular microorganisms and called moulds. Each filament is called hypha. Hyphae could be divided into a chain of cells by the formation of transverse wall, septa. As the hyphae grow and branch, they form a mass called mycelium. Fungi reproduce by forming spores in the case of sexual reproduction or by forming chlamydia, asexual reproduction. Fungi which are unicellular forms, spherical or ovoid, and do not form mycelium are called yeasts. They reproduce by budding. Okay. Look at this picture. Here we can see moths. Moths, they are present in the form of the hyphae. Hyphae it's like a branch structure, and it became to grow and create a mycelium. Mycelium and mycelium it has on its surface exospores, and exospores is necessary for reproduction. For reproduction, if if the septa of the if heifer contains septa, if heifer is uh, not like a one cell with a lot of nucleus. If all cell have its own place with the septa, with the shell, we call that kind of fungi high fungi. And if fungi don't have septa, if their gyphy is look like one big cell with a lot of nucleus, we call them lower fungi. Lower fungi without septa in their gyphy. And mycelium are of three kinds. Vegetative mycelium are those that penetrate the surface of the media and absorbs nutrients. Here we can see vegetative part of mycelium. It is necessary for getting nutrients from the substance, from the media. And aerial hyphae, it's a reproductive part of the mycelium for growing, for reproduction, for multiplying, and like that. Reproduction of fungi can be sexual or asexual. Sexual requires the participation of two genetically dissimilar fungi of the same species. So it is necessary two cells of the fungi, and typically occurs in three stages of steps. Plasmogamy. Plasmogamy involves the joining of the protoplasm and required the decomposition of the cell wall, separating the hyphae involved. Karyogamy. Karyogamy involves the joining of two haploid nucleus and results in formation of diploid cell called a zygote. Haploid cells nuclei have only one set of chromosomes, while diploid cell nuclei have two. Meiosis. Meiosis is a process involving separation of chromosomes and division of the diploid nucleus into two haploid parts. And the important feature of meiosis is the formation of new genetic combination, and not possible through asexual reproduction. So for sexual reproduction, it is necessary to have two cells with the haploid genome, haploid nuclei, and then they are joined together, and their nucleus of one cell and nucleus of second cell are joined together. Joined together make a diploid nucleus. Then it divides into two into uh, so then it divides into two haploid parts, and we have, and in the result, new fungi with new genetic combination create. Asexual reproduction occurs via division of nuclei by mitosis, which the absence of meiosis. Other mechanism associated with the nuclear cycle result in recombination of hereditary properties and genetic variation. So by mitosis we have creation of new cell, and in this process take part only one cell. Reproduction in fungi, asexual, only mitotic cell division. Somatic can be yes by budding, modes by hyphae fragmentation. So one part of the hyphae give a new life. Also asexual spore. Asexual spore is a sporangia, chlamydia spores, and conidia. And sexual is meiosis. Sexual spore, oospore, zygospore, ascospore, and basidiospore. Zygomycota mainly. Okay, now we will talk about classes of fungi which have medical importance. So Zygomycota mainly saprophytes. Saprophytes means they are not parasites. They are not parasites, and they can cause an infection, but it will be very extraordinary. It is normally for saprophytes to live in the environment and not to use the host, not to use the human or animal for as a host. Coenetic hyphae have no crossbones, many nucleus within a single plasma membrane, so they are lower fungi, and also they have haploid nuclei, have a dormant form, the zygospore. Mucor and black bread mold rhizophus are the example of Zygomycota. Look here. Asparangium is a structure inside which the spore develops. It's held aloft on an aerial hyphae called the sporangioform. So this is sporangium. Sporangium it's a kind of asexual spores. Asexual spores they create on the end of the hyphae, and they are like endospores. So they are inside the sporangium. After breaking down sporangium, they became free and go to find a new media to create a new fungi. And Bucor, it's one of the example of Zygomycete, and look here we can see sporangium. This is closed sporangium in the closed and not broken sporangium, and inside this sporangium there are a lot of sporangiospores for multiplication, for reproduction. And after breaking down the shell of sporangium, a lot of spores became free and goes to uh, to create a new life, new fungi. Okay, this is mucor. And next, Ascomycota, production of haploid ascospores through the meiosis of diploid nucleus in a small sac called anascus. Asexual reproduction be the production of airborne spores, conidia, includes some include 30,000 species. For example, Aspergillus, Trichophyton, Microsporium, and yeast. And they also, Ascomycota, they have, as we see on previous slides, they can have sexual and asexual reproduction. Okay. Here we can see kind of spore which are called conidia. Conidia, it is different from sporangia because sporangia is inside the sporangium, but conidia is outside, like a flower. And conidia also asexual spores born externally on hyphae or on the conidiophore. Conidiophore is the specific hyphae that have on its end conidia. And here we can see conidia spores. One of the example of Actinomycetes is Aspergillus, because and Aspergillus it can cause different infection, like infection of the skin, infection of the respiratory tract. Usually it causes very difficult, very strong pneumonia in the case of immune comprehensive people. And this genus include Aspergillus fumigatus, Aspergillus niger, and other are commonly associated with the infection in immunocompromised individuals. The conidiospores formed by these fungi are small, readily airborne, and easily breathed in. Various species of Aspergillus have also been found to cause deep or systematic mycosis when introduced through several tissue trauma associated with lawnmower and chainsaw accidents. Okay, and yeasts. Yeasts are unicellular fungi. Unicellular fungi, they also belong to the Actinomycetes, and their way of reproduction, the main way of reproduction of yeast are budding. It's an asexual process, and it creates by a cell division. Cell divide to the two cell, and usually the daughter cells stay on the surface of the maternal cells. That's why they have their shape. Alcoholic fermentation, they use alcoholic fermentation to obtain energy. That's why their bioproducts carbon dioxide and alcohol, and that's why yeast can be used in the food production and like that. It's a very simple biotechnology, domestic biotechnology to use yeast in production some food. Reproduction of yeasts. They use budding to reproduction. The parent cell produces one or more projections called buds, which later develop necessary structures and detach to grow into new individuals. Here you can see bud. Then this bud became to grow and became a new individuals. Fission in this process, the parent cell splits in two equal halves, each of which develop into a new individual. And this is Candida albicans in the infected tissue. It's a Deuteromycetes. Candida belong to the Deuteromycetes, fungi unperfective. Candida albicans is a type of yeast-like fungus commonly encountered as an inhabitant of the human body, forms part of our normal flora. Candida population are usually kept in check by bacteria competing with them for space and nutrients. When bacteria population are reduced as occurred during treatment involving antibiotics, these fungi can reproduce rapidly and cause tissue damage resulting in disease symptoms. Candida is commonly associated with thrush in the mouth, vaginal and eye infection, but in severely compromised individuals, this fungus can cause potential fatal septicemia. Usually in HIV infection, Candida can cause very serious systematic infection with septicemia, infection in the bloodstream resulting in disease symptoms. So here you can see Candida in electronic microscopy, and Candida is use asexual way of reproduction, also by budding. Here you can see new bud and new individuals on the maternal cell. Next is Basidiomycota. They use asexual reproduction, occurs much less frequently, produce basidiospores in basidiocarp, includes 25,000 species, and one of them is Cryptococcus neoformans. And Cryptococcus neoformans is also Actinomycetes, yeast-like fungus, commonly associated with bird dropping. Spores from this fungus gain access to the body through the respiratory system, enter the bloodstream, and migrate to brain, where they can cause potential fatal form of meningitis. Cryptococcus like Candida is most dangerous to severely immunocompromised individuals. Usually, HIV infection is associated with Cryptococcus. Okay. Most fungi are saprotrophs and not damaging to other organisms, so they don't cause the infection. But some fungi parasitize plants or animals, and they cause mycosis, fungal-induced disease occurring in humans and other animals. Mycotoxins and intoxication. Various types of fungi produce toxic substance, collectively referred to as mycotoxin. Ingestion of fungus toxin can cause intoxication, poisoning due to ingestion of toxic substance, sometimes results in reduced liver function, hallucination, or death depending on the type of fungus involved. In addition to being pathogenic and produce of toxin, many fungus release spores that are among the leading cause of atopic allergy, high fever, and humans. For example, Aspergillus, it is very allergic fungi, so it can cause very strong allergic reaction. Okay, that's all.